Good morning, children. Welcome back to botany class. Today we will discuss about sexual reproduction in flowering plants, that is chapter 2, part 4. Last class we have discussed about the male reproductive structure, that is the pollen grain. Now we will discuss about the female reproductive structure, that is the gynecium. It contains the units called pistil or carpel. One pistil or carpel contains three parts. First one, the stigma at the tip region. It is the landing platform for the pollen grains. Second part, style. It is an elongated tube-like structure connected with the stigma and the basal bulged portion called ovary. So the pistil or carpel contains three parts. What are they? Stigma, style and the ovary. Ovary contains the ovules. Inside the ovary is the ovarian cavity or locule. The placenta or the small ovule is located inside the ovarian cavity. Arising from the placenta are called megasporangia or ovules. The number of ovules and the arrangement of ovules or the placenta we already studied in the last classes. So the ovules become the seeds. Let us discuss about the structure of an ovule. See that one? Now we are going to study about the structure of an ovule. The ovule is a small egg-like structure attached to the placenta, or the stalk of the ovule is called funicle. See that one? The stalk of the ovule is called funicle. The body of the ovule fuses with the funicle is called hilum. The first part is called stalk of the ovule is called funicle. The body of the ovule and the funicle is joining portion is called hilum. Hilum represents the junction between the funicle and the ovule. Each ovule has one or two protective envelopes or the coverings are called integuments. See that one? One protective layer or the two protective layers are present normally in a seed. So that is later it becomes the seed coat. So maybe the integuments, that is called the integuments, the coverings, outer integument and the inner integument. That are the coverings of the seed, that is giving to the protection of the seed. These integuments covering the entire ovule except at the tip, where there is a narrow opening is called micropyle. Micro means small. Small opening is there. Only that portion, remaining all the portion, the integuments or the coverings are present. It is the end of the pollen tube during the process of fertilization. See that one? Now what are the parts we studied? We listen that one. The micropyle forms one end of the ovule. The remaining, the other side, the opposite to the micropyle end, the chalacal region is present and it is the basal part of the ovule. Basal means that is the starting part of the ovule. At the chalacal region, funicle and the integuments joins to the nucellus. that is the central region of the ovule, that is made up of a diploid, colorless, thin-walled parenchymatous cells containing food reserve materials. In the nucleus of a mature ovule contains the embryo sac, that is a female gametophyte, which develops a megaspore. This megaspore is formed inside the nucleus. Normally, an ovule has a single embryo sac formed from a megaspore through the reduction division. Reduction division, another name, meiosis. Now we will see one more time the structure of the ovule. First part, it is called the funicle, that is the stalk of the ovule. Second, hilum, it is the region between the funicle and the body of the ovule. Then the integuments, covering of the nucleus, outer integument and inner integument. Then the fourth portion, nucleus, that is the thin-walled parenchymatous cells nourishing the tissue that covers the embryo sac. Nourishment, giving to the nourishment of the embryo sac. Then the chalazal region, that is the basal portion of the nucleus where that integuments will arise, and the micropylar region, small opening present at the tip of the integuments. Funicle, hilum, integuments, nucleus, embryo sac, chalazal region and the micropylar region. So these are the parts of the megasporangium or the ovule or the female gametophyte. Now we will discuss about the megasporogenesis. Megaspore means that is the ovule, genesis means the formation. See that one, or the megasporogenesis or the female gametophyte or the embryo sac. In embryo sac, where it is present inside the nucleus structure. Nucleus structure, that is the nourishment tissue of the embryo sac. That is first, megasporogenesis, the process of formation of megaspore from megaspore mother cell by the process of meiosis is called megasporogenesis. The process of formation of the megaspore from where megaspore mother cell, through which process, meiosis. Meiosis is also called the reductional division. So automatically 4 megaspores will form. The process of megasporogenesis occur in the megasporangia or the ovules present inside the ovary. Each ovule has outer integuments enclosing the nutritive tissue is called nucleus. Some of the cells of the nucleus develop into a diploid archisporial cells. What is that archisporial cell? The plant cell that divides to give rise to a sporocyte and is part of sporangium, or it is a primitive cell, or it is a group of cells, is called archisporial cell. Now we will think about how to form an ovule. See that one, inside the nucleus structure, the embryos are how it is forming. First formed cell, it is called the archisporial cell. Now we'll think about any type of the fruit. Fruit is the outer covering and inside the fruit contains the seeds. Now we are discussing about the seed, that is the ovule. Ovary develops into the fruit, ovule develops into the seed. Now first, megasporogenesis. First we are discussing about the archisporial cells, that is a group of cells present in the nutritive tissue, nucleus cells. Division to form the and forming a sporogenous mass of tissue during the division, and the sporogenous cell develop into megaspore mother cell. This megaspore mother cell divides by meiosis to produce tetrad of haploid megaspore. Tetrad means 4. 4 haploid megaspore. See that one, mitosis means equational division. 2 cells will form. And the meiosis means it is the reductional division. Haploid 4 cells will form. Now here also from the megaspore mother cell 4 haploid megaspores will form. Out of 4 megaspores, usually 3 megaspore cells will degenerate or disappear, and 1 megaspore cell at the micropylar region enlarges to become a functional megaspore that is called embryo sac. So listen, from the megaspore mother cell, 4 cells are forming. 4 haploid megaspores will form. Out of 4, 3 will degenerate, and only 1 cell is the functional megaspore that is called the embryo sac. This type of development is called monosporic development. Mono means from the single spore development, megasporic development. Now we will discuss about the megagametogenesis. It is the process of formation of egg cell from a functional megaspore. Why it is called monosporic? Because 4 megaspore will form, in that 3 will degenerate, and only 1 is the functional megaspore. So they are called monosporic development. It is the megagametogenesis, means it is the process of formation of the egg cell. Gametes are the egg cell from a functional megaspore, or now the functional megaspore is called embryo sac. Okay. The nucleus of the embryo sac divides mitotically. Mitosis means 2 division cell to form how many nucleus? 2 nucleus. See, listen here. From the embryo sac, 1 cell divided into form 2 nuclei which move to opposite poles of the nucleus. That one megaspore or the embryo sac cell, they are forming into 2 cells, and it moves to the 2 nuclei, moves to the opposite poles. Again, 2 more successive mitotic divisions will be there. 2 more means the 2 cells are formed. Divides to form mitosis to form. There are the 4 cells. 4 cells again divide to form 8 cells. Then occur how many nucleus? After the results of the divisions, they are forming 8 nucleate stage. Now both the side 4 nucleus cells are present. Now the arrangement, we will see that one. 3 nucleus in the cella cell end surrounded by a cell wall and are organized to form 3 antipodal cells. See that one, out of 4 megaspore cells, they are 3 nucleus from the megaspore cells. 1 is the functional one. In that first mitotic division, second mitotic division, third mitotic division, 8 nucleus are forming. In that 8 nucleus, 4 in the cella cell region, 4 in the micropylar region. In that 3 cells in the each region, in the cella cell region, 3 nucleus are surrounded by a cell wall and organized to form the antipodal cells. Now next it is 3 nuclei in the micropylar region surrounded by a cell wall and organized into 3-celled egg apparatus. In that 3 cells, 2 consist of the synergids and 1 egg cell. See that one? 3 cells in the cella cell region, 3 cells in the micropylar region. Then that 2 cells are remaining, both the side. 2 nuclei from each pole move to the center to form the polar nuclei present in a large central cell. So the mature embryo sac is 7-celled and 8-nucleated. See that one? The mature embryo sac is 7-celled and 8-nucleated. I think you are, and hope you are understood that the process of the arrangement of the cells. See that one? The embryo sac or the female gametophyte, the arrangement. Listen one more time. That is that near to the chalazal region, 3 cells are the antipodal cells, and the micropylar region, 3 cells are forming the egg apparatus. In that egg apparatus contains 2 synergates and one is the functional cell. And from the both side, out of 4 cells, 4 cells, one cell moves to the central and it is forming to the polar nuclei, that it is called one cell. The 2 nuclei is considered as a one cell. The chalazal region 3 cells, micropylar region X contains that the egg apparatus contains 3 cells and in the central cell contains the 1 nucleus. So they are called 7-celled and 8 nucleus is present. Okay, now we will see that one again one more time. I am explaining about the distribution of the cells within the embryo sac. Listen, 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus consists of 2 synergates and 1 egg cell. What is the synergates? The synergates have a special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in the guiding the pollen tubes to the synergate. See that one? The egg apparatus contains 3 cells. What are the 3 cells? 2 synergates in the side and the middle the egg cell is present. The synergates have the special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in the guiding the pollen tubes into the synergate. These 3 cells are at the chalazal end are called 3 cells at the chalazal end are called antipodal cells. The large central cell is mentioned, that is called as 2 polar nuclei. Thus, a typical angiosperm embryo sac at the maturity, that are the 7-celled 8-nucleate cell. See that one? The first we should think about that one, the structure of an ovule, Megaspore Genesis. Then the arrangement of the egg apparatus. Okay, that are the an embryo sac and the angiosperm embryo sac contains the 7-celled 8-nucleate. Thank you.